Hello everyone, this is Alberto at the Buckler Lab, and today I'll be talking about GWAS in this Tassel tutorial video series. So this is also going to be multi-series videos. So the first one, this is very conceptual. This is sort of just to remind you what a GWAS are and what we're doing and sort of the options we have in Tassel. And then I'm going to cover the specific models in depth in additional videos. So what are GWAS, or genome-wide association studies? Basically, they are models for detecting statistical association between genotypic and phenotypic values. In other words, we want to know which regions of the genome associate with specific traits or differences in specific traits. And the trademark of these studies are these Manhattan plots, which basically show on the x-axis, like in this example, the 10 chromosomes of maize, and the y-axis shows a measure of statistical significance. In this case is the negative log 10 p-value, and you want to see something like several peaks, very discrete peaks, which show you the regions that are candidates for association, and the number of peaks depends on the statistical power, but also the number of genes underlying your trait. So this is sort of what you will expect to see. And what's the underlying model? So if we have a p-value, it means we made a hypothesis test. And here I have two examples for exemplifying what's happening. So I simulated data, I simulated height for an individual of whatever species you want to think about. And we have two different SNPs, the one on the left and the one on the right. And these SNPs have genotypic values, but the individuals that have these genotypic values are different. So for SNP1, you have the individuals that have AA are on average shorter than the individuals that have one copy of T, and those in turn are also shorter than the ones that have two copies for T. So you can see that this is associated with height, and when we do a regression on this, it's going to show a significant p-value, and that's something that we're going to plot, and that's going to be our Manhattan plot. And the example on the right is the opposite, where you have no association, basically whether individuals have A, T, or the heterozygote. We are not going to see that. So this is the additive model that we're going to be testing. There's additional models for dominance effects, but this is the first model. If you're interested and you want to revise a little bit more the literature, go into your quantitative genetics and you can understand a little better. This is just a primer of the underlying model. However, life is not as simple as that. There's things that happen and we have to consider before doing GWAS, and one of them is population structure. And here I have an example of dogs. So let's say you have the question, what genes control height in dogs? So you basically say, oh, okay, so Chihuahuas are small and Great Danes are big. So you go, you sample several Great Danes and several Chihuahuas, you measure them, you genotype them, and then you run a model. So here is pretty obvious that something's going to happen. Yes, you're going to see the genes that are associated with height in your results. However, everything else that is clearly different between these two breeds is going to show significance, and it's going to be a false positive because it's not truly associated with your trait, but actually also associated with the other traits or this population structure that underlies these samples. Hopefully most of the time you're going to have some recombinants, some more random mating populations, and we can infer this population structure from the genotypic data. Tassel has PCA, or principal component analysis, implemented, and this example is from the humans, and you can see this is a map of Europe, and from genotypic data you can see that there is a lot of structure such that individuals that are geographically closer related tend to be genetically more closely related. So this makes sense, and we can use this information as covariates for GWAS to control for this non-random mating that is happening, and you can also estimate how much variance this is explaining in terms of your phenotypic variance and your genotypic variance. And I'm going to cover this in a special video down the road. So another consideration is that even after you have control for this population structure, you have what we call hidden relatedness, and for that we use the mixed model. And here, for example, I have an European royal family tree. So this is just to exemplify the idea of like hidden relatedness is all those relationships of cousins, second cousins, third cousins within a specific population. Even if you say, okay, I have control for people who are coming from Spain and people who are coming from Germany, people who are coming from Spain, you may have first cousins and second cousins who, on average, are going to look more similar than to people who are completely unrelated. And we can also estimate a matrix that captures that relatedness in the form of a kinship matrix and use that in our model as a random effect. And I'm going to talk about random effects and mixed models in a specific video. So this is just to remind you what the consideration is and how to solve them. And this is implemented in Tassel. A third consideration is the missing data in terms of both genotypes and phenotypes. And one thing you can do is imputation. Imputation means that you take information surrounding your SNPs, and in this case I'm using just Sudoku just to give you an idea of how, even though there is values that are not present, you can use the information surrounding these present values to actually infer the true value. And you can do that with genotypes with certain accuracies, but you can also impute with certain confidence some phenotypic values. The discussion of whether you should impute or not phenotypic values, I'm going to discuss it further. Maybe you don't want to include that individual, maybe you want to impute it. But we have methods implemented in Tassel to impute genotypes with very good accuracy and very, very fast. And we have considerations that we're going to suggest about missing data in case of phenotypes. Another consideration is rare alleles. And most of the time you're going to deal with this by filtering, which is also implemented in Tassel, and I have already covered some filtering options in previous videos. And basically what this means, and this plot is also showing simulation data with the same number of individuals than in the previous example, and what you see is that we only have 2 observations of the individual with the homozygous T allele, and we only have 4 for the heterozygote. So in this case, it's really hard to know whether the T allele is having a significant effect for height or not. And even though I sampled a distribution which had higher mean height, you can't actually tell because of variance. And what's worse is that you may actually get an outlier. You may get that in reality these individuals should have a mean similar to your regular allele, your major allele. But if you have one observation and by environmental effect, it's significantly higher or lower than the other values. If you have this outlier, it's going to drive association that is going to be a false positive. So you want to take that into consideration, and filtering is going to help you deal with these issues. Another issue that comes up in GWAS is the multiple testing. I'm not going to go into the theory, but basically under the null hypothesis, p-values are uniformly distributed, which means that you're going to have values that are less than typical significant threshold of, like, let's say 0.05. So you're going to have 5% of the times things that are not associated with your phenotype pop up as significant. And you want to take care of that. The analogy I make is with children. If you're driving and you ask the question, are we there yet? So most of the times you're not there, but if the kid asks enough, you're going to say yes. And this is just sampling a random distribution of replies to the child. And even though that's false, you are normally not there. You just made a false positive just because the question was asked too many times. So the same happens with statistical data. If you ask the data too many questions, you're going to have something like that. And there's things like Bonferroni or false discovery rate that can help you make a decision. And we're also going to look at QQ plots in the GWAS videos, sort of to diagnose to what extent this is very relevant and where you can observe it and what you can do about it. Finally, a very important consideration is that we do not have whole genome sequence data. We have high density genotyping, but we don't have the causal polymorphism. So the hypothesis we're testing, the model, is actually on tag SNPs, SNPs that are highly correlated with the causal SNP, but that are not causal. So that means that if you have an association, the tallest, the highest, the most significant p-value may not be the actual causal polymorphisms. And the way to know which polymorphisms around could be related or could be actually causal, you can look at LD around it. And in Tassel we have implemented LD methods, and this plot is, for instance, using Tassel. And this is going to help you understand the underlying association and see candidate genes that surround according to the LD structure. So some general conclusions from this video. First of all, your experimental design is going to dictate the power and the results. So there's power in numbers. Think about the experimental design before you decide to do GWAS. Think about the marker density you require, and think about the number of observations you require according to your phenotype. Measure heritability, and know that GWAS, once you have the data, it's an iterative process. You're going to be running models several times, and you have to also look at the data, look at distributions. So it's not a one-click, I'll get the result sort of deal. Proceed with caution. Always be very critical and reasonable, and use your intuition of the biology, of the statistics, to sort of guide your decision-making, your filtering, and hopefully your interpretation of the results. And really do your homework, go back to your quantitative genetics and look at it. There's several reviews online in Nature or like other journals that can help you understand the GWAS world. And finally, a Manhattan plot is only the beginning. Even if you have a Manhattan plot, you have to do downstream analysis and really keep an open mind and be ready to explore the exciting world of the association of genotype and phenotype. So that's it for now. Thank you very much for watching. Next videos are going to be the actual GWAS models. And see you there.